All right, so we just finished talking about Mendel's laws, law of dominance, law of independent assortment, law of segregation. But not everything follows Mendel's laws. Mendel's laws always follow complete dominance. Okay, those were all of the P crosses followed complete dominance, which means that if a dominant allele is present, it will be shown. However, not all characteristics show complete dominance. Sometimes the hybrid or the heterozygote is a combination of the two. So in this example, we have a red flower and a white flower, both homozygous. And when you cross them, you get a pink. Now, this sounds a lot like the blending hypothesis that we discussed prior. However, the difference is if in the blending hypothesis, if you took this hybrid and you blended it with another hybrid, you'd get a variation on pink. Like everything would be pink, but it would be some kind of variation of that. But because we have the incomplete dominance, what will end up happening is some of them will indeed be pink. You can see right here. Okay, some of them will indeed still be pink, but because each one of them has a dominant allele, we are going to have some that might be red and some that might be white. And that's the difference between the old blending hypothesis and incomplete dominance. If you have an incomplete dominance problem, which you will practice, they will tell you that the heterozygote or the hybrid looks like pink. All right? Now, that does not at all affect the genotype. So the genotypes would be the same. Homozygous dominant red, homozygous dominant white, heterozygous pink. So the genotype portion will be the same. Another difference is codominance. So codominant is when two alleles, if they are present, are both expressed. And our blood typing is an example of that. So we actually have 4 different blood types, and this is shown on the chart. This is complicated. You don't have to know it all. All you need to know is that if you have blood type A, you can either have 2 A alleles or 1 A, and then they just put an I right here. I like to put an O just so that's not confusing. So you can either be homozygous A or you can have a heterozygote with an O, or with nothing there. I is a nothing. B, same thing. Now, because A and B are both dominant, you can also have an AB blood type where both A and B are expressed. And because A and B can have this heterozygote condition, you could also have O blood type, okay? Now, the blood type has to do with the antigens present on the outside of your red blood cells. So if you have A blood, you have like this spike. If you have B blood, you have this circle. And then if you have both spikes are on your blood type. If you have none of them, if you are homozygous recessive, then you're O and you have nothing on your red blood cells. And if you take anatomy, we'll get into this. We'll even type your blood, see what's going on there. So it's really cool. So let's look at this. Maria has type O blood. So we know Maria has to be OO because the only way to have O blood is homozygous recessive. Her sister is AB. All right. Her sister is AB. Now, they know that their maternal grandparents are type A, which doesn't really matter. What are the genotypes of the girl's parents? Okay. So you can actually do a Punnett square, even though this is not parents, these are kids. You can actually do a Punnett square backwards, and you can see that the way that this happens is that the parents, you would have one parent who was an A that was a heterozygote for an A, O, and you'd have the other parent who's a B who's heterozygote. That way, the Maria can get these 2 O's from both of her parents, and her sister could get the A and the B from both of the parents. Okay, so I kind of did a backwards Punnett square, but you might have some of these to do. They will tell you, they might not say co-dominant, they may say they are both dominant or they are both shown when present. Okay, so you might have a couple of these to do in your practice. All right, the next one I'm not going to give you any to do because it's so complicated. There's the next two. Pleiotropy is one. This is where one gene is going to influence a multiple characteristics. And we already talked about sickle cell when we discussed mutations, but sickle cell, if you have the mutation in your germline, in other words, in your sex cell, can be inherited. When this gene is messed up, then it changes the shape of your red blood cells. Now, if you don't have sickle cell, you have, so sickle cell and non-sickle cell can be co-dominant. But here's the difference. So in sickle cell, oh, I took out the picture. So in sickle cell, your cells are going to look like this, right? There'll be all, if you have the trait for sickle cell, you're homozygous dominant, you'll have all your cells look like this. If you have normal, then all your cells are going to look like this. And if you're like this, then only half of your cells because it's co-dominant. Now here's the deal. This one gene of sickle cell also has an influence on your resistance to malaria. So if you are this guy, if you are heterozygote for sickle cell, you actually have an increased resistance to malaria. So that's one gene, but it affects two different traits, and that's pleiotropy. Now, an additional one is polygenetic. Poly means many, and genes, G-E-N-E-S. All right, so many genes affect one characteristic. And this characteristic, it's usually an additive effect, right? So every single gene codes for either a different aspect or slightly changes the gene prior to it. And human skin color is an example. I won't actually have you do any of these because as you can see from this picture, it's super complicated. You can have very light or very dark skin. We start with that. Then you have these you have gene A, you have gene B and gene C. So depending on if you're homozygous or heterozygous for each one of these traits, you're going to fall anywhere on the spectrum of shades of skin color. And it's actually even more complicated than just these three genes, but they kind of simplified it for you here. So this is extremely complicated. I'm not going to ever ask you to do one of these, but you do need to know the terms polygenetic and pleiotropy. Those are terms that you will need to know. So, but I'm not going to ask you to do that on anything. All right, I want you to answer your checkpoint question. This is something that we're going to talk about in another video, traits being affected by environmental factors. So we're going to talk about that in a different video later. All right. Chromosome theory of inheritance states that genes occupy a specific loci, that's a vocabulary word, or space on a chromosome, and that chromosomes undergo segregation and independent assortment in meiosis, which they do. We already know that. So now, this is not a different theory against Mendel's laws. This is actually kind of proving Mendel's laws. But this does give us an idea about this picture is showing you essentially this is meiosis 1 and meiosis 2 and how the chromosomes are separating independently and going in different sperms, not paying attention to what the other genes are doing. And we've already discussed that in previous episodes. All right. So now, but what we found out is that some genes are very often inherited together, which would seem to disprove the law of segregation and independent assortment. But what we know now is that they're actually on the same chromosome linked together, okay? Or very close together. And I think they have an example here. All right, so if you look at your regular old prediction right here, 9331. This would be everything sorts independently, nothing's linked. You should see a 9331 ratio, right? We know that because we did dihybrid crosses. But when they did the crosses in real life, this is what they got. So what they realized was that this long pollen and purple flower are close together on the chromosome. All right. And so I'm just going to show you this gene map really quick because the idea is this. This is for flies traits. So you have maroon eyes, black body, cinnabar eyes, vestigial wings means very small wings that don't work, brown eyes, and this is a chromosome. Now, because of crossing over, hold on, let me go back to crossing over. Let's look here. So remember crossing over, right, where these chromosomes exchange the genetic information in meiosis. Because of crossing over, right, we get a lot of genes that get recombined, okay? And we call that recombinant. Sorry. And so the recombinant is something that happened when genes were crossing over. So crossing over between homologous chromosomes makes new combinations of alleles. All right. Linked genes can be separated, right? However, the closer they are together, the less likely they are to cross over. Closer together, the less likely they are to unlink, we'll say, or recombined. All right. So if we look at this genetic map, that I showed you a minute ago, we are going to see that the frequencies between how linked some items are on the same chromosome will have to do with where they are. In other words, if I'm looking at this, look down here, brown eyes is way down here and maroon eyes is way up here. I bet those recombine a lot. They're very far away on the map. But I bet you that vestigial wings and down curved wings or vestigial wings and cinnabar eyes are almost always inherited together because they're so close together. So I'm not going to ever ask you to do a chromosome map or anything like that, but it's important for you to understand that when crossing over occurs, new traits are inherited together as a result of that recombination, and we call them recombinants. And the more often that occurs is when they're further apart on the chromosome.